In the last video, we talked about simple diffusion across the membrane, whether it was diffusion of solutes from an area of high concentration to an area of low concentration that the membrane was permeable to, including the diffusion of water from the area of high water concentration, which is the same as a low solute concentration, to the area of low water concentration, which is the same as a high solute concentration. But there are lots of things that can't diffuse directly across the membrane that our cell needs to transport across the membrane. So we're going to talk about the other ways that molecules can move across the membrane. The first one we're going to talk about is called facilitated diffusion. Facilitated diffusion is when a protein channel allows for the diffusion of molecules down their concentration gradient. So this is still diffusion. The proteins go from the area of high concentration to the area of low concentration. A protein just helps or facilitates the diffusion of these molecules. This is going to be really important when we discuss the structure and function of our neuronal membranes in chapter 48 next. Some of these proteins are always open. They're just a channel or a hole that makes the membrane permeable to these solutes all of the time. But others can open and close and therefore control when and how the membrane is permeable to particular solutes. So facilitated diffusion is still a form of passive transport. It doesn't require an input of energy. Molecules just diffuse from their area of high concentration to their area of low concentration. But sometimes you need to force things to go against their concentration gradient. Doing this requires an input of energy, and that's called active transport. So active transport moves things from an area of low concentration to an area of high concentration, making it even more concentrated on one side of the membrane. And this requires a source of energy such as ATP. The example that we're going to go through is the sodium-potassium pump. It's going to be essential for how our neurons function that we're going to discuss in the next chapter. So for our cells, the concentration of sodium and potassium on either side of the cell membrane are different. So in the extracellular fluid, the concentration of sodium is high and the concentration of potassium is low. Vice versa, on the inside of the cell, the concentration of sodium is low and the concentration of potassium is high. So that's the first thing you just need to memorize. You need to know that sodium has a higher concentration outside of our cells and potassium has a higher concentration inside of our cells. The reason this is true is because of the sodium-potassium pump. The sodium-potassium pump is a transmembrane protein that does active transport. It uses energy from ATP to force more sodium out and more potassium in. So the sodium-potassium pump has a binding site for 3 different sodium ions. And when the sodium ions attach, it allows that protein to become able to be phosphorylated by ATP. When ATP phosphorylates this protein by adding this charged phosphate group, the protein folds into a different shape. It flips so that it's now open to the extracellular fluid. And in this shape, the sodium ions are released and the protein has a high affinity for potassium ions. So potassium ions, even though they're at a low concentration outside of the cell, are attracted to this protein. And when the potassium ions attach, the phosphate group breaks off and the protein folds back into its original shape. Now it's open to the inside of the cell. And in this shape, the protein has a low affinity for potassium and it releases potassium into the cell. So as this cycle continues, what you guys need to know is the sodium-potassium pump is a form of active transport. It pumps sodium out of the cell, and specifically it pumps 3 sodium per turn out of the cell, and it pumps 2 potassium per turn into the cell. And what this does is create these concentration gradients where sodium is high outside of the cell and potassium is high inside of the cell. And those actually drive our neuronal impulses. Another form of active transport is called cotransport or coupled transport. It uses the energy of ATP to set up ion gradients, and then those ion gradients actually drive movement of molecules across the membrane. So in this example here, this is a sodium-glucose coupled transport. The sodium-potassium pump pumps sodium out of the cell, and so energy was used to set up this concentration gradient. Now, molecules want to diffuse down their concentration gradient from an area of high concentration to an area of low concentration. So sodium will naturally diffuse back through the membrane if there is a permeable protein that allows sodium to move into the cell. So in this case, this co-transporter binds sodium ions that want to diffuse into the cell and glucose molecules that the cell wants to bring in. Even if there's lots of sugar in the cell wants to bring more sugar in. And so by using the energy of the sodium ion concentration gradient, it can force glucose into the cell. So again, this is another example of active transport. In addition to these proteins pumping small molecules or ions across the membrane, we also have bulk movement across the membrane. And when we rearrange the whole membrane, this requires an input of energy as well. So we've already kind of discussed exocytosis, things being released from the cell when vesicles fuse with the membrane and release their contents. Rearrangements of the membrane that fold the membrane in also require energy and are a form of active transport. So endocytosis brings things from the outside into the cell. So again, these large bulk rearrangements of the membrane are a form of active transport. There's different kinds of endocytosis. So when large solid particles like bacteria or food particles are brought into the cell, this is called phagocytosis. Thinking back to chapter 6, do you guys remember what cytoskeletal component pushed the plasma membrane out, making these little pseudopods? Okay, those were actin microfilaments. So actin microfilaments support these structures, the pseudopods, that eventually wrap around and make a food vacuole that brings the food particle or a bacteria to the lysosome. So assembling those proteins and pinching the membrane together requires a source of energy. When the cell builds these little cup-like structures to bring solutes and small molecules into the cell, that's called pinocytosis or cellular drinking. And then the last example I want you guys to know about is called receptor-mediated endocytosis. So there are receptors, proteins, on the surface of the cell that bind specific small molecules. And the generic name for the thing receptors bind is called a ligand. What happens when these receptors bind their ligands is it attracts proteins on the cytoplasmic side of the membrane that causes them to fold inward and make these little protein-coated vesicles. What this allows for is a high concentration of a specific molecule being brought into the cell. This is different than phagocytosis or pinocytosis, which just grabs whatever's in the area. Okay? So some other small molecules can come in, but we get a high concentration of our specific ligand brought in this case. So one example of this is the LDL receptor. LDL is low density lipoprotein. It's the bad cholesterol. And to metabolize it, receptors have to be able to bind it and bring it into the cell. But if you have a mutation in your LDL receptor, either the part that binds LDL or the part on the inside of the cell that interacts with the proteins that fold it in, this can lead to high familial cholesterol.